Assalamualaikum and hi everyone. Today I am going to share with you guys about the latest finding published in 2019 about genome sequencing by BMC Genomic. The title of the journal is Genome Sequencing Provide Insight into the Evolution and Antioxidant Activity of Chinese Bayberry. Chinese bayberry is an economically important fruit tree that characterized by its juicy fruit that rich in antioxidant compounds. The Myricaceae family comprises three genera, of which Myrica, comprising approximately about 50 species, is the most widely distributed across the warm humid regions of Asia, Europe, America, and South Africa. In addition to producing flavoral fruit, different organs of Chinese bayberry are used in traditional Chinese medicine. Chinese bayberry extract contains antioxidants against inflammation, allergies, diabetes, cancer, bacterial infections, diarrhea, among other health issues. Myricanon induces apoptosis in HEPG2 liver cancer cells through the generations of reactive oxygen species. It also has anti-cancer effect on the HeLa, which is cervical cancer, and PC3 that known as prostate cancer. Myceritin exerts selective cytotoxic, pro-apoptotic, and anti-metastatic effect on prostate cancer cells by inhibiting PIM1 and disrupting the PIM1 interaction. Chinese bayberry leaf flavonoids and proanthocyanidin will inhibit the growth of the ovarian cancer cell line. Chinese bayberry, also known as a treatment of human gastric cancer cells, with cyanidin-3-glucose markedly increased. It is an important tumor suppressor gene inactivated in many human cancer. Flavonoids, including cyanidin-3-glucoside and anthocyanidin, may have beneficial effects on diabetes by reducing oxidative stress in muscle and fat. Extract from Myrica rubra show protective effects on liver injury and non-alcoholic and alcoholic-induced fatty liver disease, which may be due to its potent antioxidant properties. Thus, major flavonoids, anthocyanidin and anthocyanin of Chinese bayberry leaf have a strong chemical and cellular antioxidant activity. Flavonoids are abundant in Chinese bayberry fruits, but the genetic basis of their biosynthesis remain unclear. The sequence, active sites, and expression level of the genes related to the antioxidant signaling pathway in Chinese bayberry remain unknown, but elucidating them would be useful for the development of a new medications. So, in this part, there are several results that have been made from the experiment conducted. The figure A shows a microcollinearity analysis of Vitis vinifera, Microrubra, and Malus domestica. This region represents one collinear set highlighted in orange. And for the figure B, navy blue rectangle indicates the predicted genes in the genome. One gene in Vitis vinifera corresponds to one gene in Myrica rubra, whereas one gene in Myrica rubra has two homologs in Malus domestica. For the figure C, the single peak of the Myrica rubra paralog indicates that there are no recent whole genome duplication except for the polyhexaploidization event, which is common to eudicots. For comparative genomic analysis, a five-way comparison showed that 4,074 genes family were shared by the five species and 712 genes family were unique to the Myrica rubra, while a four-way comparison revealed that 4,270 genes family were shared by these four species and 891 genes family were unique to the Myrica rubra. The classifications of specific genes in the above two classes was similar in different genes family. This indicates that these functions were enhanced during Chinese bayberry evolution. The antioxidant signaling pathway of flavonoid, anthocyanin, and terpenes in Myrica rubra comprises genes of the above-mentioned four groups. and it is suggested that they are enhanced in Marica rubra in comparison with other plant species. For gene analysis of antioxidant and pharmacological activity, the antioxidant activity of Chinese bayberry extract and its ability to inhibit cancer cell reproduction are linked to the total flavonoid, anthocyanin, and terpenes. We found that the fruit of zoysia have a relatively high total flavonoid content and that their antioxidant activity decreased during the course of development. In the flavonoid pathway of Chinese bayberry, shikimate or hydroxycinamolyltransferase, or known as HCT, was present in high copy numbers. Its main role is transferring acetyl groups from p-coumaryl-CoA and caffeol-CoA to shikimate and quinate. The expression level of HCT homologs during fruit development were generally low. Thus, the high gene copy number and the specific active residue of each gene copy might not be correlated with high content of flavonoid in Chinese bayberry fruit. There are two enzymes that play important roles in anthocyanin synthesis, which is anthocyanidin 3-O-glucoside 5-O-glucosyltransferase and anthocyanidin 3-O-glucosyltransferase. Low expression levels were detected and the expression level of 4 copies were negatively correlated with the decrease of total flavonoid on total antioxidant capacity during fruit development. It is because it is highly expressed throughout the ripening process and it may contribute to the accumulations of anthocyanin during fruit ripening. High copy number and the variations on the functional residue contributed to the high anthocyanin content in zoysia fruit. The expression pattern of genes related to flavonoid biosynthesis indicated that the pathway is active in Marica rubra during fruit ripening. The high expression level of SCOMAT may have contributed to the high flavonoid content in zoysia fruit. Chalcone synthase functional residue of Marica rubra have high similarity to those in the other 5 species. In Marica rubra, chalcone synthase was highly expressed during the early stage of fruit development. It suggesting that this gene is important for the high total flavonoid content and total antioxidant capacity of young fruit. There are few biosyntheses that may be important for the total antioxidant capacity during the fruit development. Most Marica rubra genes in the antioxidant signaling pathway have multiple copies via tandem duplication event, and many genes contain structure and amino acid changes of functionally active residue with low similarity. In this part, 8 methods were carried out in this research. These are the methods. Firstly, genome and transcriptome sequencing. Fresh leaf of 10-year-old Myrica rubra zoysia were collected and the fruit were not visible to the naked eye until the 10th day after pollinations. For the transcriptome sequencing, fruit samples were collected 10, 20, 30, 40, and 50 days after the initial pollination event. Samples were denucleated, ground immediately, frozen in liquid nitrogen, and stored in an ultra-low temperature. Secondly is a DNA extraction. Genomic DNA was extracted using the settled trimethyl ammonium bromide DNA extraction protocol. And thirdly is genome assemble and annotation, which the novel assemble pipeline was developed for a genome assemble. A whole genome shotgun assemble was used to generate context. Fourth, comparative genomics, where all against all comparisons of protein were performed using the basic local alignment search tools such as BLAST. The evolution of gene family size in Marica rubra and related species was evaluated based on the maximum likelihood phylogenetic risk with estimated branch length. And the fifth, flavonoid contents and total antioxidant capacity of Marica rubra fruits. Each Chinese bayberry fruit sample was dried to a constant weight and ground into a fine powder, and then they will be calculated such as stated. For RNA extraction, transcriptome sequencing, and assemble and annotations, the total RNA of Marica rubra fruit was separately isolated from 50 samples using triazole reagent. The integrity and concentration of RNA were checked. 15 RNA sequence library were constructed and then sequenced. The clean RNA sequence data were then mapped to the genome assembled to perform reference guided mapping. And then for the homologue of genes and protein sequence, homologues of genes involved in flavonoid and terpenoid metabolic pathway were performed using the neighbor joining methods of MEGA software, which also known as molecular evolutionary genetic analysis. The protein structure homology modeling was performed. And then, lastly, is a quantity real-time PCR. Complementary DNA sequences were obtained from the total RNA samples using the FastGain real-time kit with gDNA, and then a PCR was performed. This paper reports a comprehensive genome sequencing study of Chinese bayberry and the transcriptome analysis of antioxidant pathway in Chinese bayberry fruit. The results obtained provide a basis for functional genomic research and genetic improvement of Myrica rubra, and are valuable for inferring evolutionary relationships across related species.